Hello, and thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Abood. I am the science editor at the Human Vaccines Project, and I will be your moderator today. We've commented on many grim milestones throughout the COVID-19 pandemic, and now there is yet another, as the US reached 1 million deaths due to SARS-CoV-2. The highest death toll of any country in the world, despite widely available access to vaccines and antiviral medications. This once unthinkable outcome occurs as multiple subvariants of Omicron circulate across the world. BA4 and BA.5 in South Africa, and BA2 and BA2.12.1 in the US. These highly transmissible subvariants are causing a spike in cases that is probably severely underreported due to the use of at-home testing or the presence of asymptomatic cases. This has many scientists urging the US government to prioritize the development of a new generation of COVID vaccines, including those administered intranasally, which may have the potential to block infection as well as severe disease and death. In the meantime, the US FDA just authorized booster doses this week for children ages 5 to 11 who have previously received 2 doses of the Pfizer-BioNTech COVID-19 vaccine. Before we get started with today's presentation, I wanted to announce that the application link for the 2022 Michelson prizes is now open. These next generation grants are awarded annually to support promising early career investigators who are working to advance human immunology, vaccine discovery, or immunotherapy research. The Michelson Medical Research Foundation, together with the Human Vaccines Project, established these $150,000 prizes in 2017. More information on the prizes, as well as a link to the application portal, are available on this slide and on the Human Vaccines Project website. So please check there for more information or to apply. It is my pleasure to now introduce today's speaker, Dr. Ripley Ballou, a senior scientific advisor at IAVI, who leads their work on developing broadly neutralizing antibodies for HIV prevention. Dr. Ballou has a long history in vaccine development, including serving as vice president and head of GSK Global Vaccines US Research and Development Center. Prior to that, he was vice president and head of clinical research and translational science at GSK Vaccines. Ballou also served as deputy director for vaccines at the Bill and Melinda Gates Foundation. He trained in internal medicine and infectious diseases and began his work on vaccines at the Walter Reed Army Institute of Research, where he led the team that with GSK co-developed RTS-S, the world's most advanced malaria vaccine. Dr. Ballou received his MD from Emory University School of Medicine and completed his residency and fellowship in infectious diseases at Walter Reed Army Medical Center. During today's presentation, please send me your questions using the Q&A function in Zoom. Feel free to use the chat for general comments, but please don't post questions there. I will ask our speaker a broad selection of your questions after the presentation, and we will have about 20 minutes for discussion. With that, it is my pleasure to introduce Dr. Rip Ballou.
Thank you, Kristen. It's really a pleasure to be here. For full disclosure, in terms of conflicts of interest, at the time that I retired from GSK in very early 2020, I was not working on COVID vaccines. The company began working on them just at the same time that I was retiring. I did obviously go to IAVI, and IAVI has its own program, internal program, initially working with Merck on COVID vaccines, and is now working on an intranasal vaccine based on the VSV platform. I also early during the pandemic served on the as an advisor to Moderna's clinical development program and advised the board on clinical development issues early in the development of their COVID-19 vaccine. It's really a pleasure to be here, and I don't know the participants very well, but I assume that, like myself, most of you who have been working on COVID vaccines were probably working on other vaccines when COVID hit. And it's obvious to me, and I'm sure it's obvious to you as well, that we would not have been able to make the progress on COVID if we had not been making the investments in novel technologies during the pre-COVID period. And the point of my discussion today is to emphasize how many of these activities that have enabled COVID vaccines of many different flavors came from technology due to investments in HIV, malaria, and TB. Very challenging diseases that required new ways of thinking about vaccine development. And many of the technologies that have now been used successfully for COVID vaccines really had their foundation in these other diseases. And I'm going to point that out as we go through it. I'm giving this as an update on where we are with HIV, malaria, and TB vaccines, but where relevant, I'm going to try and make a link to how this has informed what we've done with COVID. And of course, the learnings from COVID are going to help us in these difficult diseases, and I'll try and highlight those as well. So I'm going to share my screen here and begin this presentation. And Kristen, can we see that? Everything's good? Okay.
Thank you, Rip.
Great. All right. I've tweaked the title a little bit here, but I just want to remind everybody, HIV, malaria, TB, problem not solved. We have, in the case of HIV, 680,000 deaths in 2020, 1.5 million new cases each year. In malaria, 240 million cases of malaria, 670,000 deaths, mostly in children living in sub-Saharan Africa. And TB, the one that we don't really talk about so much, 1.5 million deaths, and many of those had HIV co-infection, 10 million new cases in 2020. So the problem is with us big time still for each of these diseases, and we desperately need effective vaccines. I'm going to start with HIV vaccines, and my overriding message here is that this has been a proving ground for new technologies, and there has been a paradigm shift in the strategy, which has been driven by progress in structural biology. And those of you that have been working on COVID vaccines know how important that last issue is. So HIV, it's probably the most challenging vaccine target that's ever been attempted. There is enormous genetic diversity. There is persistent infection of cells at privileged sites. There is high level of glycosylation of the envelope glycoprotein, which is referred to as a glycan shield that interferes with access of antibodies. And particularly neutralizing antibodies, the antibodies that can actually neutralize this virus are structurally shielded by this glycan. And neutralizing antibodies are only rarely elicited during natural infection. Now, it may be that up to 50% of people that are infected begin to develop these, but very, very few of them actually go on to develop mature, broadly neutralizing antibodies that actually can control the infection. This is what referred to as so-called elite controllers. Much of that is being done by these very rare broadly neutralizing antibodies. And to date, we've not been able to do this by vaccination. So this is really the immunological challenge that we have here. Now, on the plus side, studies in the Shiv-Rhesus model of HIV reveal that neutralizing antibodies that bind to discrete conserved epitopes on the trimer can block infection in vitro and in vivo. And these are rigorous challenge models that can demonstrate that it's possible to do this if you have the right antibodies. And in addition, the cellular immune arm is also really important. Effector CD8 T cells directed against important HIV epitopes can kill HIV-infected targets. And while there is a huge focus today on how do we induce broadly neutralizing monoclonal antibodies by vaccination, there is in parallel a recognition that we will need the cellular arm of the immune system triggered, and that may require multiple vaccination strategies put together. Now, there's been a lot of work in HIV. It's one of the most well-funded of the big three over many, many years. But unfortunately, the results to date of large-scale trials, whether they were phase 3 or phase 2B, but with large numbers of volunteers, have been very disappointing. There has only been a single study that has shown a glimmer of efficacy, and that was the so-called RB144 study done in Thailand, and attempts to reproduce that study with more modern technologies have not been successful. I would point out on this slide, though, that you'll see recombinant adenovirus, canary pox, the use of DNA vaccines, the use of Ad26, the use of adjuvants have all played a big role in getting these particular candidates to large-scale trials. And in the process of doing that, this has been very helpful for informing the regulators about how to approach these vaccines and what to think about in terms of safety, and have really informed the field. And I think that as you know, a number of these technologies were employed for COVID vaccine development. Now, the current generation, I'm going to call it second generation of HIV vaccine design, it's really been made possible by a revolution in structural biology. And this has yielded critical new tools that have allowed us to understand what the native HIV trimer looks like and how antibodies bind to them. And so, things like cryo-EM, MPEM, other technologies that have really allowed the ability to understand the structure, to go in and to technically genetically modify the structures to make them stable, and which, as was done with the spike protein for SARS-CoV-2, really had their beginnings in work done on HIV, and in parallel at the same time, work done by the VRC on influenza and RSV, all of which have informed and provided tricks that the geneticists and molecular biologists can use to go in and stabilize these very complex proteins that typically exist in multiple forms, and there's only one form that we actually want as an immunogen. And if you can't stabilize it, you can't make a good immunogen. So this work was begun in the early 2000s and progressed, and by 2013 actually had an approach that you could articulate to say, this is a way we could use these tools to actually make an HIV vaccine. And again, this approach was to map the binding of human broadly neutralizing antibodies. These are antibodies that have been recovered from long-term non-progressors, mostly from people both in the United States, but also from people living in sub-Saharan Africa, and looking at how these antibodies bound to native light trimers using cryo-EM and X-ray crystallography. Obviously, you had to have the native light primer to begin with, but that then allowed us to really take it to this next step here, which is identifying these broadly neutralizing antibodies, mapping them in great detail on the HIV envelope, and then being able to design an immunogen that is based upon the epitope, the structure of the epitope, and this has been accelerated by the use of a really interesting nanoparticle approach that uses ferritin derived from Helicobacter, which does not cross-react with human ferritin, and that gives you a structure that you can display on a nanoparticle-like protein, these very interesting immunogens that we hope can trigger these rare BNAb precursor B cells. And the idea behind this new approach, which we call germline targeting, is to tickle these BNAB precursor B cells and encourage them to develop into the broadly neutralizing antibodies that we would like to have in large quantity in the blood to protect against an HIV infection. So the work that Bill Schief and folks at the Scripps CHOV-D have been working over the last decade have allowed them to design an immunogen that they refer to as EOD, that's engineered outer domain germline targeting version number 8, EDOGT8, that is germline targeting, that is able to recognize these precursor B cells that have the capability to evolve into broadly neutralizing antibodies. The idea is that once you have elicited that initial population, you would come in with a second immunogen that is a little more, it's more closely related to the envelope, also being presented as a nanoparticle that would allow you to expand these memory B cells, and finally to come in with a third step, which is really a native-like trimer for polishing, and with what's called a polishing step. And while this process, this is sequential immunization of at least 3 different vaccines, a very different approach to vaccination than we have tried with any other disease, to my knowledge. This germline targeting, the goal here is to enhance through somatic hypermutation enough of these plasma cells so that you actually have circulating BNAbs. And this work was modeled in mice. It was shown to work in non-human primates. And most recently, we've actually had proof of concept of this approach for a particular kind of broadly neutralizing antibody called a VRC01 class response that binds to the CD4 binding site. This has been now demonstrated in humans. This IAVI G001 that Bill Schief and his colleagues ran and will be published shortly, demonstrated that it was safe and well tolerated. It was given with AS01B, the GSK adjuvant that is used in not only its malaria vaccine, but its Shingrix vaccine for herpes zoster. Was used in this, and it was able to generate not only strong antibody responses, but strong CD4 binding site responses. There was high positivity. Essentially, all but one of the volunteers produced BRCA1 class responses. And here, we're not actually measuring neutralizing antibody, we're actually sequencing B cells that demonstrate that they are of the class that have the potential to become broadly neutralizing antibodies, and they're characterized by the use of VHA 1+2 and 5 amino acid long CDR3 component. So high frequency of VRCO class responses, and really now the next step is, can we take that step and actually boost? Now, this study was done with recombinant proteins and adjuvants. It's a very effective way of immunizing. However, it takes about 2 years to make one of these proteins. And if you can imagine doing this now all with protein-based work, you're looking at several decades of work to get an answer, really. And that's, we find that to be a big challenge for HIV. So we are working with Moderna to understand whether mRNA, as has been so successfully used for COVID-2, can be COVID-19. Can this be used to prime and essentially recapitulate what we saw with a protein boost and a protein adjuvant, and then boosting with the core 60-mer? This is a study that's ongoing right now. The preclinical data were very promising, demonstrating that you can express these ferritin nanoparticles and express a complex immunogen in mRNA and boost them. But now we're actually doing this in the clinic, and this will be extended to our our African sites in IAVI 003, where in Rwanda and Republic of South Africa and Kenya will be doing these studies. Finally, mRNA, can these be used to deliver native-like trimers? And this is being done in HPTN 302. So here's an example of where the success of mRNA in COVID-19 vaccines has really has the potential to accelerate the work that we're doing in HIV. And finally, there is a question about, can we simplify this strategy by, instead of focusing on these in particular immunogens, can we do this by focusing on the trimer? The challenge here is that these trimers have an immunodominant base, and that's demonstrated as the red area on this base view in the center of this picture. That base is exposed because it's not covered by the glycan shield, and most of the antibodies that are directed against the base are non-neutralized. A number of quote stabilized HIV trimers have been made, but in vivo they undergo degradation, and this enhances more off-target binding. So the challenge is in using this whole trimer approach is to really direct the immune response against these privileged sites where the neutralizing antibodies can bind, and not get boosting of these off-target antibodies that they're not harmful, but they don't help, and they just essentially outcompete the neutralizing responses. And there's been very recently a very interesting paper from the Duke Chabdi that has shown that using mRNA engineered trimers displayed again on ferritin nanoparticles, they look very promising in preclinical studies, and a vaccine based on this is now in GMP manufacturing and heading towards clinical trials. So this may provide a complementary way to the way that the Scripps Chabdi group is doing to try to get to a vaccine that can induce these broadly neutralizing responses. So, how can progress with COVID-19s, and my view is that they will accelerate HIV vaccines. mRNA, the amazing thing that's happened with COVID-19 mRNA vaccines is that we have a safety profile at a scale that would have never been possible had there not been a pandemic. That's one of the pluses of this huge exposure of the population. We understand that there are some adverse events that are rare, but we have much better understanding that these can be given safely. We've seen that you can use these to engineer highly complex, finely tuned immunogens, which is exactly what we need for HIV. The ability to manufacture quickly and at large scale is expanding globally. And so, the issues around access could potentially be addressed with this mRNA platform. There's important information about the durability and the requirement for boosters that will inform how we think about vaccine development for HIV. And there's evidence that the LNPs in of themselves are self-adjuvanting and actually promote follicular T-cell engagement. And this is really an important aspect in terms of what we're trying to achieve with HIV vaccine. On the engineered viral vector approaches, the demonstration that both the chimp adeno and the Ad26 platforms can be scaled to meet global demand. In fact, Ad26, that Johnson & Johnson's vaccine for COVID, would not have been possible had they had not made the investment in Ad26 manufacturing to support their HIV program. There has been, of course, a suggestion of a class effect of a safety signal regarding clotting disorders at risk subjects, and this is useful for people that are thinking about these kinds of platforms for other vaccines to be aware of. And we have seen that they are suitable for use in heterologous prime boost regimens if we need that to complement an antibody response with a CMI response. And on the adjuvanted recombinant proteins, these are not yet in large scale use, but I would note that there is now the introduction of a fourth potent adjuvant, matrix M, to complement the availability of GSK's AS01, and AS03, and the MF59 adjuvant. Really, a decade ago, these adjuvants were not available or were only being used in experimental studies, and now we have lots of safety data on these, which is really helpful. Also, I think the recombinant proteins provide additional support for the use of nanoparticles to present complex images. So a lot of new information and complementary reinforcing information coming from COVID vaccine development, which is going to help HIV vaccines. Now I'm going to turn to malaria vaccines. This is an area a bit more in my expertise. This is an area where adjuvant protein subunit vaccines dominate the landscape. And we've been able to take advantage of a human experimental challenge model for a number of decades, which have really helped drive progress in this field. In the area, when we think about malaria vaccines, we have to consider the life cycle. And although a number of different candidates and approaches have been contemplated, the work that is progressing most successfully is really based upon 3 targets. One of these is a target that is actually the form of the parasite that is injected by the mosquito and infects humans, and that's called the sporozoite. And this is the most successful vaccine for malaria, RTSS, AS01 GSK's vaccine, which was recommended for general use in 2021 by the WHO, is an infection blocking vaccine. In other words, pre-erythrocytic, before you get this parasite into your blood, it blocks the parasite from getting into its initial target cell, which is the liver. There are several other approaches that are also targeting this. I'm going to speak primarily about RTSS because there's some interesting data here to consider. There are groups that are working on the blood stage parasites. Here, the real problem is that when you're talking about trying to block or shut down an established infection with malaria, you're dealing with a huge number of merozoites circulating in the blood, thought to be on the order of 10 to the 12th parasites at any one time in a person that's clinically symptomatic with malaria. But there is an interesting vaccine called RH5, which is being used with various adjuvants that targets a very conserved protein on these blood stages, and at least in initial clinical trials, looks like it has the ability to reduce the rate of replication of the parasites, which could be important in modulating clinical disease. And finally, there's an approach that we call sexual transmission blocking vaccines, which target the sexual stage of the parasite. This is where the male and female gametes actually are taken up during a blood meal by a female mosquito and are fertilized in the gut of the mosquito and go on then to develop into sporozoites in their salivary gland. And there are several very interesting antigens that show quite good preclinical promise. Again, very complex antigens to produce, and potentially some of the lessons from COVID might help this field. I want to talk a little bit about the role of what we call controlled human malaria infection. We know that human challenge models of COVID are being looked at right now, and these may give us a lot of information about the pathogenesis of COVID and potentially clues to new vaccine development. This was done because there is no good animal model for P. falciparum. And while more recently there has been work with humanized mice, and when we began this work back in the 80s, those were not available. And we were able to use a human challenge model to really run through a lot of what we thought were promising clinical candidates, take them to the clinic, see if you can protect against malaria infection, and if they didn't, stop that program and move on to another. And this worked. This requires, of course, a highly standardized process that uses parasite detection methodologies that allow you to stop an infection with chloroquine treatment early on in the before people actually get very ill. This model, it's been standardized by using the bites of 5 falciparum sporozoite infected mosquitoes. It can be adapted to blood stage vaccine development by actually injecting a small number of malaria infected red blood cells, and this has been done safely and successfully by a number of groups. And this has allowed us to down select candidates to go into the field. We can do field efficacy studies in malaria because there are high rates of natural infection. So CHMI has been really helpful to help us distinguish between one vaccine versus another in early phase 1, phase 2 proof of concept studies. It's been very helpful in selecting formulations and working out schedules and looking at potential improvements in vaccines, working out mechanism of action, and doing systems analyses on why is it that some people are protected and others are not. Field trials, these studies, these CHMI studies have been primarily done in malaria naive adults. Field trials have been primarily done in malaria experienced populations, starting with adults, progressing to older children, and then younger children for phase 3. Now, the RTSS vaccine is a recombinant protein virus-like particle, a nanoparticle that uses the hepatitis B core antigen as the element that produces the virus-like particle. It consists of a section of the circumsporozoite protein that has not only antibody but T cell targets. Part of this is fused to hepatitis B surface antigen. The other is not fused, but when they are co-expressed in yeast, they self-assemble into this virus-like particle. Work on this began in 1997, where a proof of concept study showed that 6 out of 7 volunteers were protected in a CHMI study in US adults. This progressed to studies in sub-Saharan Africa in 2003 that led to a phase 3 in 7 African countries in 2009. Based on the data that came out of that in 2015, we had EMA approval under the Article 58 procedure, and subsequently a pilot introduction in almost 800,000 children in Ghana, Malawi, and Kenya. And in 2021, based upon safety data and new data that came out showing that this vaccine used with seasonal malaria chemoprophylaxis can show up to a 70% reduction in malaria mortality. WHO has recommended its general use in children from 5 months of age. So really made a lot of success in malaria, but it's not a perfect vaccine. And this may be familiar to those of you that have been working on COVID-19. We have an issue with durability and the requirement for boosters. I'm showing you here a series of figures that came from the phase 3 program. The panels on the left the panel on the left upper portion of this are from children aged 6 to 12 weeks of age. In other words, this is a birth cohort that was immunized, and a second cohort in this phase 3 trial was children from 5 to 17 months of age. And if you notice, shortly after vaccination, we get very high levels of antibody, but these antibodies decline in a biphasic fashion. And where the initial decline in antibodies shows a half-life of about 6 to 7 weeks, and then it tapers off, and the half-life subsequently is around a year. And the antibody responses to the malaria component of this are very similar to the antibody response to the kinetics of decline of the hepatitis B component. So we don't think this is due to the fact that there's something funny about the malaria antigen. However, the important point is that it takes very high levels of antibody to get high levels of efficacy. And if you look at this curve under E, the top right, you can see this modeling shows that in order to get about 50% of efficacy, you need somewhere between 80 and 90 international units of antibody per ml, which is a lot of antibody. That's a pretty whopping dose of antibody if you relate that to micrograms per mil. What I've shown on all three graphs with this red dotted line is where that predicted 50% efficacy shows up. And you can see that for much of the time, in the absence of boosting, and that's no boosting on the first two panels, top and bottom, and boosting on the second and the middle panels, you can see that you can boost those antibodies back up, but they drop back down. And the overall, when you now follow these children out to 4 to 5 years, the overall efficacy that is being followed here is on the order of 40 to 45%, which is important given how many malaria infections occur every year. But if you look at what happens right after, in that very short time afterwards, the efficacy can be as high as 70 or 80%. And so, you know, this is again something which I think recapitulates what we're seeing with the mRNA vaccines. We get good effective boosting, but the requirement for, I mean, of the initial course of vaccine, but the requirement for boosting is important. What we are not dealing with in malaria is the evolution of the parasite. So it's not under immune selection and evolving from an antigenic point of view the way SARS-CoV-2 is. There are several other approaches. There's a vaccine from Oxford called R21M, which is using the matrix M that Novavax is using for their COVID vaccine. It is a very similar vaccine to RTSS, uses a slightly different structure that has a higher density of repeat epitopes, and that may allow for a lower dose to be used. There's been some promising vaccine efficacy, and phase 3 is planned, not yet recruited, but this could help provide additional sources of vaccine, because the vaccine supply for the RTSS vaccine is going to be limited for a number of years. I mentioned the RH5. This is also being studied with matrix M, and an attenuated sporozoite approach by Scenaria. You can get high level of protection against CHMI in US adults, but vaccine efficacy in malaria exposed individuals has been disappointing, especially in young children. And there's work ongoing to better understand why that is. Now, turning to TB, we have a licensed TB vaccine, BCG, but we need something better for sure. This slide talks about the natural history of tuberculosis, and identifies places where an improved vaccine could actually help. Exposure to TB is primarily coming from adults and adolescents with active tuberculosis. And one of the primary populations that are being infected are infants and young children. This is where BCG is being used globally to control the TB burden. It works pretty well in preventing disseminated tuberculosis in young infants, but when it's given as boosters, it's not so great. And when it's given to young adults and adolescents, doesn't seem to change the course of tuberculosis. So new vaccines are needed. And there are a number of places in the pathogenesis of tuberculosis where these vaccines could be used. Now, BCG is an old vaccine. It was derived by serial passage of M. bovis back in 1921 and has been given at or close to birth. More than 3 billion doses have been given and is generally considered safe. It has good efficacy against severe TB disease in young children and infants, but efficacy wanes by adolescents, and revaccination studies have shown conflicting results and have had no impact on overall mortality. Consequently, there is an improved BCG that has been genetically modified to improve its immunogenicity by replacing an enzyme that facilitates the release of mycobacterial antigens in the cytosol. This is currently in phase 3. It's called BPM 1002. This is being studied in the Republic of South Africa and also in household contacts of MTB infected persons. And finally, another replacement vaccine for BCG called MTBVac. It's a live attenuated vaccine, which is derived directly from a virulent MTBV isolate, where specific attenuation of two important virulence genes has been done to essentially render this an avirulent but very authentic otherwise Mycobacterium tuberculosis immunogen. It maintains a superior repertoire of potentially protective antigens versus BCG, because many of these antigens were lost during passage and attenuation of BCG. It has superior efficacy versus BCG in mice, guinea pigs, and non-human primates. There have been no safety signals observed during phase 1 and phase 2 studies in adolescents, infants, and adults. It is entering phase 3 for prevention of TB disease in infants later this year. And there are ongoing phase 2 studies for prevention of TB disease in adolescents and adults. Now, this vaccine comes with a cost. It may be a better MTB vaccine, but it interferes with the current standard interferon gamma release assay, which is one of our most important diagnostic tests, because it actually, MTBVac retains antigens that are used in the test to demonstrate interferon gamma release, indicating positivity. So if this vaccine is successful, we may actually require new diagnostic assays, which will add to the complexity. And finally, there have been some interesting data with recombinant proteins and adjuvants. GSK's M72 AS01E, it's another version of AS01, the same version of the adjuvant that's being used in the malaria vaccine. This is a fusion protein consisting of two MTB-derived antigens expressed in E. coli, given with a strong adjuvant. A phase 2b trial of about approximately 3,000 individuals demonstrated 50% efficacy over 3 years. Now, this was in MTB-infected HIV negative adults. And so this is a vaccine that you could give to people that have already been infected, but who are at risk of developing active pulmonary tuberculosis, which is not only not good for them, but it spreads the infection in the community. So this vaccine has been licensed to the Gates Medical Research Institute, who are planning and leading a phase 3 program. And there's another similar approach called H56 IC31, again, a recombinant protein and an adjuvant from the Stoughton Serum Institute called IC31. Is currently in phase 2 in patients successfully treated for pulmonary TB who are at risk for recurrence. So progress is being made. This is my last slide, and I just want to again summarize some of the things that, in my opinion, have come from the vaccines for HIV, malaria, and TB that have informed how we think about the development of COVID-19 vaccine. So nucleic acid vaccines, DNA and mRNA, were studied first in HIV before they were studied in COVID. We know, we've seen from human challenge studies that when they're used smartly and correct and safely, they actually can help you in the development of new vaccines. And this may be very interesting for nasal vaccines. The acceptability of complex adjuvants that drive difficult to achieve immune responses, the challenges of durable protection and the importance of booster doses, the use of adenovirus vectored vaccines and heterologous prime boosts. How do you deal with equitable access? We know half the world has yet to receive, particularly in Africa, has yet to receive a COVID vaccine. We have the same issues here in HIV and malaria and TB. Genetically attenuated live vaccines that induce robust immune responses. We know that there's work like this going on in COVID as well. Importance of vaccines designed to prevent infection, not just modulate disease. Approaches to stabilizing structures of complex antigens, and the importance of using structurally correct immunogens to begin with. One of the things I like to say is you cannot fix a bad vaccine with a good adjuvant. If you don't have the structure right, it's not going to work. We've learned this the hard way in HIV, and it's really helped the COVID community develop effective vaccines. So I'll stop there. I want to thank all of the donors that make so much of this work possible and that support our work at IAVI. I'll turn it back to Kristen. I hope I haven't overgone the time frame here.
No, well, I mean, it's unbelievable that you could cover those 3 areas so quickly if you think about it. So thank you so much for that excellent presentation. It's a lot to think about. And I'm wondering, because, you know, actually one of the questions I was going to ask you was about adjuvants. And I appreciate your comment on you can't fix a bad vaccine with a good adjuvant. But what role do you think adjuvants might play in boosting durability? Maybe for SARS-CoV-2 vaccines, but probably likely for HIV and other things where you mentioned that, of course, the durability of the immune response is going to be really critical.
Yeah, you know, I think what we have seen is that in the absence of replicating vaccines, which the durability issue there is very different. Once you have a replicating vaccine, and measles is like this, yellow fever, you get very, very long immunity. And why? Because there are small reservoirs of the immunogen that are tickling the immune system. But if you don't have that solution available, then the curve, the decay curve that you are seeing following vaccination, repeated boosting and making sure that the timing of the boosters is important in getting a maximum response. But once that's been achieved, there's only so far you can push the immune response, and it's going to self-regulate, and there will be a decay. So I think that where adjuvants could play a role is by pushing that initial level of antibody much higher than you might be able to achieve, for example, with mRNA vaccines. We haven't really seen good head-to-head studies on this, at least. I mean, there's some suggestion when people have taken serum samples that you get, you know, orders of magnitude different antibody response. And I do think that by getting those higher levels of antibody, you are going to affect the kinetics of that decay and hopefully minimize the need for boosters. We're just going to have to see, and I hope that we have the opportunity to look at combinations of these vaccines to see what sequences make sense and how to minimize the complexity of delivering these vaccines over time. My own personal belief is that COVID's going to be with us for a long time. We're going to need boosters that are more closely attuned to the circulating viruses, and we want to maximize the response when we actually give one of those vaccines.
Yeah. Yeah, and to that point, we've obviously seen that the uptake of getting people to take booster doses in the US is somewhat of an issue. So when you look at the complexity of the HIV vaccine regimens that may be what are required to actually induce protection, what do you think about the practicality of those? I mean, it's obviously been called very elegant science, but do you see it as an actual product that can be developed and deployed?
I mean, if it actually takes, I mean, I didn't go into it in this detail, but if you actually think through that, there could be as many as 9 or 10 different immunogens that need to be given in a specific sequence to induce the kind of response that we think is necessary. If that's what's required, I don't think it'll ever see the light of day. I mean, you can do the studies, but as a practical approach, it's going to be very difficult. Now, I think what we do know is that you can combine immunogens, and it needs to be demonstrated experimentally, but there are plenty of vaccines that are very successful today that contain a large number of different immunogens, and that work. So unfortunately, in order to do this development, you have to do them individually, but really the next step is to see what can we put together. And, you know, maybe the Duke approach of working with a trimer as opposed to the individual immunogens might help simplify this process. And the two approaches may work together. Right. So I think we have to keep an open mind about this and not say, oh, it'll never happen. I was told that malaria would never happen, and here we have a vaccine now recommended by WHO.
That's right. That's right. That's absolutely right. So following up on that, we have a question on whether you can comment on data assessing the ability of RTSS to prevent infection rather than clinical infection across all age groups. And does RTSS or R21 type vaccines have any role on other use cases like pregnancy or elimination where a high degree of protection against infection would be required?
Yes. So, I mean, the mechanism of action of RTSS is that it prevents infection. We know that. That's what all of the controlled human infection studies show. Either you're infected or you're not. And it's the power of that challenge model, because it allows you to discriminate and understand it's either one or the other. Your feet are up or your feet are down. It's not a disease modulating vaccine. The thing to realize in the field is that these kids are being infected every day. Every day a mosquito's coming along and potentially infecting somebody. So it's very difficult to get that clean demonstration of protection in a field setting. It's not that it can't be done, but it's just not practical. Certainly not in a phase 2 or phase 3 study. Now, the question about can this vaccine be used in other populations, other use systems, for sure. And that's what was really elegantly shown by Brian Greenwood and the team. I alluded this very quickly, where they use this in Burkina Faso, which has very intense seasonal transmission, and not just young children, but young adults are also really key. I mean, everybody gets infected, but particularly younger people are more infectious to mosquitoes. And so it's in that setting where historically people have used seasonal chemoprophylaxis to shut down malaria transmission during the period when the mosquitoes are biting, and then they take it away during the rest of it. But this study compared seasonal chemoprophylaxis, vaccination alone, or the combination of the two. And vaccines the RTSS performed almost the same as seasonal chemoprophylaxis alone. But when you use the two together, you saw this 70% reduction in mortality, which is huge. And this is an example of, once you have these vaccines, you figure out how best to use them. And the use of these vaccines in other settings certainly has been contemplated. The issue with pregnant women is a little bit different. There, it's, yes, you want to prevent them from becoming infected, but the morbidity and mortality is really related to the exquisite sensitivity, the blood stage component of it during this period. So I think the short answer is yes, but you have to do the studies.
Yeah. Okay. And so we had one other question on whether you saw a path at some point for a combination malaria vaccine that targets across all 3 life stages.
Well, I think if we had, I think where this makes perhaps most sense is in conjunction with what we call transmission blocking vaccines. I think if we can block, if we can reduce the infectivity of the parasite for mosquitoes and reduce the risk of getting infected once if the mosquito is infectious, that could have an impact in a community. We're not there yet with the transmission blocking vaccines. These are very, very complex antigens. They've only been tried so far to be produced by recombinant protein technologies. They have lots of cysteines. They form difficult structures. This may be an area where mRNA vaccines could help reduce the manufacturing complexity of these immunogens. So I think that that a multi-stage vaccine approach, it's been thought about for years. We actually did one years ago, but none of the components worked as well as they should. They were given in a canary pox vector, but it does make sense. And hopefully, once we get better immunogens for these other targets, we can look at combinations with a pre-orthocytic vaccine.
Right. Right. That's very interesting. And we had a question on the difficulty of testing the HIV vaccine candidate regimens with the long-acting PrEP on the horizon, and how the complexity of conducting even the trials.
Yeah, I mean, this is something that the community is having lots of conversations about. Obviously, long-acting antiretrovirals are going, if they're widely available and accessible and safe, and we don't have the emergence of drug resistance, they are going to be a central component of HIV management, and we're not contemplating stopping those so you can do an HIV vaccine trial. Rather, and the community developing broadly neutralizing antibodies is dealing with the same issue. We need to be thinking about different kinds of trial designs, so-called counterfactual designs, where we use historical controls or communities that have similar transmission experiences, but who are not participating in the trial. And regulators are engaging deeply with vaccine developers to think about how to do these studies when you have other mechanisms of preventing disease that ethically you just cannot withdraw.
Right, right. Well, I'm pretty sure we could probably keep you here all day asking you questions about all of these topics. But thank you so much, Dr. Ballou, for presenting on this work across 3 very, very important disease areas. We really do appreciate you taking the time to share your insights with our audience and for answering their questions.
Sure. Well, it's my pleasure, and my apologies if I didn't talk to somebody's pet vaccine. One of these projects, it's a huge field, as you know,
Yeah.
but my pleasure to be here and share my insights with you.
Thank you so much. And thank you also to all of the attendees for participating in today's webinar and for submitting such great questions for our speaker. I just want to quickly invite you to join us for the next Global COVID Lab meeting, which will be held on Thursday, June 9th. Our speaker that day will be Dr. Ofer Levy, who is director of the Precision Vaccines Program at Boston Children's Hospital and a professor of pediatrics at Harvard Medical School. He will present on precision vaccines, lessons learned from the coronavirus pandemic. And if you're interested in more research on COVID-19, please sign up for HVP's COVID Report, a bi-weekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. Finally, please visit our website and follow us on LinkedIn, where the full webinar series is available. And with that, I'll say thank you again for participating today. Stay safe, and we hope that you'll join us again for the next Global COVID Lab meeting. Thank you so much.